Hello students, I am Dr. Nilima Kulkarni, Assistant Professor, Department of Microbiology, Modern College of Arts, Science and Commerce, Ganesh Khind, Pune. Today we are going to see glycolysis. Glycolysis is chief metabolic pathway for glucose degradation in bacteria. The meaning of glycolysis is degradation of sweet material, that is sugar. Glycis means sweet and lysis is breakdown. Glycolysis was discovered by scientists Emden, Meyerhoff and Parnas, and in their recognition the pathway is known as Emden-Meyerhoff pathway or Emden-Meyerhoff-Parnas pathway. The glycolytic pathway has 10 reactions which are catalyzed by enzymes which are present in the cytoplasm, and these 10 reactions can be divided into two phases. The preparatory phase, which includes first 5 reactions, and the energy yielding phase, which includes the next 5 reactions. The net reaction of glycolysis involves breakdown of 6-carbon sugar glucose to produce 2 molecules of 3-carbon derivative pyruvic acid. Glycolysis is seen in many bacteria, but some bacteria use other pathways such as Entner-Doudoroff pathway or pentose phosphate pathway. Gram-positive bacteria such as Bacillus and Streptomyces use glycolysis as major metabolic pathway. Microaerophilic bacteria such as Lactobacillus, which show fermentative metabolism and produce lactic acid as end product, also use this pathway for production of pyruvic acid, which is further converted into lactic acid. Gram-negative facultatively growing anaerobic bacteria, E. coli and other enteric bacteria also show glycolytic pathway. Some nitrogen-fixing bacteria such as Azotobacter crucocum shows presence of glycolysis. With these few examples of bacteria, I will like to mention that yeasts and fungi also show glycolytic pathway and they are eukaryotes. This is the first phase of glycolytic pathway, which is known as preparatory phase, which has 5 different reactions as seen in the diagram. And the second phase is known as energy yielding phase. The first phase converts glucose into 2 molecules of glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is used as starting material for the energy yielding phase, and energy yielding phase results into production of 2 molecules of pyruvate. In the preparatory phase, glucose is energized so as to allow its degradation and release of energy in the energy yielding phase. 2 molecules of ATP are invested in 2 different reactions in the preparatory phase. Reaction number 1, which is catalyzed by hexokinase or glucokinase, and as you can see here, reaction number 3, which is catalyzed by phosphofructokinase. These 2 reactions degrade ATP and ADP is produced. The phosphate is transferred on to sugar derivatives so as to convert them into their phosphorylated metabolites. At the end of preparatory phase, The fructose 1,6-biphosphate is acted upon by enzyme aldolase. This results into cleaving fructose 1,6-biphosphate into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Isomerase enzyme converts dihydroxyacetone phosphate to glyceraldehyde 3-phosphate, thus resulting into production of two molecules of glyceraldehyde 3-phosphate at the end of preparatory phase. The glyceraldehyde 3-phosphate thus formed is acted upon by enzyme dehydrogenase, and the enzyme converts glyceraldehyde 3-phosphate to two molecules of 1,3-biphosphoglyceric acid. This is a high energy compound which is used by phosphoglycerate kinase enzyme as substrate, and the enzyme brings about substrate level phosphorylation to synthesize ATP. One more reaction in glycolytic pathway involves kinase, that is the 10th reaction converts metabolite phosphoenol pyruvate to two molecules of pyruvate, and this is also substrate level phosphorylation which converts ADP to ATP. The reaction which is catalyzed by glyceraldehyde 3-phosphate dehydrogenase, that is reaction 6, involves addition of inorganic phosphate, and the reaction involves oxidation, that is removal of hydrogen, wherein two molecules of NADH plus H plus are synthesized. Let us see the first reaction in details. You can see on the left hand side of the slide the structure of glucose. This is the cyclic structure of alpha D glucose, and the enzyme hexokinase uses this as substrate. The enzyme can phosphorylate glucose at the 6th position and converts glucose to glucose 6-phosphate. Enzyme uses ATP as donor of phosphoryl group, and ATP is converted to ADP. The reaction requires presence of magnesium ions. To understand this, we will have to see the structure of ATP. Hexokinase reaction is predominantly seen in yeasts. In some bacteria such as E. coli, a more specific enzyme which is known as glucokinase catalyzes this reaction, and this enzyme uses glucose as the only substrate. Hexokinase can act on number of hexoses such as glucose, mannose, fructose, etc. This is the structure of adenosine triphosphate. You can see on the right hand side that adenine is attached at the first position of pentose ribose, which has at the 6th position 3 phosphate groups attached to it. The phosphate groups are linked to each other by phosphoanhydride bond. The phosphates are labeled as alpha, beta and gamma. In the reaction which is catalyzed by hexokinase or glucokinase, the terminal gamma phosphoryl group is transferred onto glucose so as to produce glucose 6-phosphate. This results into production of adenosine diphosphate, which has alpha and beta phosphoryl groups. You can see here that all the phosphates have negatively charged oxygen, and hence, so as to stabilize ATP, Magnesium, which is a divalent cation, associates with ATP forming Mg-ATP complex. This is the reason why in the previous slide you can see that magnesium ion is required for this particular reaction.
If you remember in previous lecture, I mentioned that phosphofructokinase is a regulatory enzyme. In the same manner, even hexokinase is a regulatory enzyme. It can regulate, it responds to various metabolites, and it can regulate the rate at which glucose can be converted to glucose 6 phosphate. In short, it can decide how much glucose will be metabolized through glycolysis in cell at any point of time, depending on availability of various metabolites of glycolytic pathway. What is the purpose of glycolysis? It is a catabolic pathway which results into release of energy, production of reductant, and also production of precursors. Okay, so when sufficient precursors are available, rate of glycolysis will be slowed. Okay, this slowing down of the rate of glycolysis is brought about by two enzymes, hexokinase and phosphofructokinase. So we have seen the reaction of hexokinase enzyme. In E. coli, alternative enzyme glucokinase, which is more specific, acts only on glucose, is also seen. Hexokinase is seen in yeast and some bacteria.
Many bacteria, instead of using enzymes such as hexokinase or glucokinase, phosphorylate sugar using phosphotransferase system. The phosphotransferase system is a transport system which is membrane associated, and it transfers phosphate group onto sugars such as glucose so as to convert them into their phosphorylated derivatives.
So phosphotransferase system is a transport system for sugars which will phosphorylate various sugars, different hexoses at the entry point into the cell. When the sugar is taken inside the cell, it is phosphorylated. So hexokinase and glucokinase will not function in such cases when during transport the sugar is phosphorylated.
Let us see description of phosphotransferase system of E. coli and Bacillus subtilis. In the right hand side of this particular slide, the diagram describes the phosphotransferase system. On top of E. coli, let us see details of the phosphotransferase system of E. coli. The phosphotransferase system consists of three cytoplasmic proteins E1, HPR and E2A, and two membrane associated proteins including a permease 2C and a subunit of E2 protein E2B. The reaction of phosphate group transfers takes place in a sequential manner. Phosphoenol pyruvate, which is a metabolite produced in glycolytic pathway, can act as donor for phosphate group. Phosphoenol pyruvate is converted to pyruvate, and this reaction is accompanied by phosphorylation of protein E1. The phosphorylated E1 protein can donate the phosphate group to the next protein HPR and results into production of phosphorylated HPR. Phosphorylated HPR in turn donates its phosphate group to protein E2A and phosphorylates it. In the last reaction, E2A transfers the phosphate group onto membrane associated protein E2B. When glucose comes in through permease 2C, the phosphate group is transferred by protein E2B onto glucose so as to synthesize glucose 6 phosphate. Bacillus subtilis has a similar system with slight variation. In Bacillus subtilis, protein E2A and E2B both are associated with the permease 2C. The reaction sequence is necessarily the same, wherein phosphoenol pyruvate transfers phosphate group onto protein, and the phosphorylated membrane bound protein will convert glucose to glucose 6 phosphate. The second reaction in the glycolytic pathway is conversion of glucose 6 phosphate to fructose 6 phosphate. Glucose is aldose, that is at the first position in the straight chain structure you can see aldehyde group present, whereas fructose is a ketose which has ketonic group at the second position in the linear form.
Okay. So now you can see this is the structure of glucose. So here you can see these are 6 carbons in the linear structure. This is the aldose group. This is the linear structure of fructose. So what is the difference? You can see here that the carbonyl group is at position 2. So fructose is a ketose, has a ketonic group, whereas glucose is aldose, has aldehyde group here. Okay. Now when glucose cyclizes, this aldehyde group gets converted to a hemiacetal, whereas in case of fructose, it results into formation of a hemiketal. So this is carbon number 1. You have seen the linear structure. Carbon number 2 had a carbonyl group. So it involves reaction between this carbonyl and the hydroxyl group at position 5. As we have seen cyclization in case of glucose, similar cyclization will take place.
In cyclic structure, you can see that glucose 6 phosphate has a 6-membered ring as seen here, and in the hexokinase reaction, we have seen the structure. It is a heterocyclic ring with oxygen as one of the member. The 5 carbons are present in the ring structure, and the 6th carbon is projected above the plane as CH2OH. In glucose 6 phosphate, this 6th carbon is phosphorylated. Phosphoglucoisomerase enzyme converts glucose 6 phosphate to its isomer fructose 6 phosphate. The reaction involves conversion of aldose to a ketose sugar derivative which has phosphate group attached to the 6th position. In structure of fructose, you can see that it is a 5-membered ring, and the first carbon is it is projecting above the plane of the ring as CH2OH, and the 5th carbon also projects above the plane of the ring as CH2OH, and when it is phosphorylated, it is converted to CH2OPO3 2-.
Now, this particular reaction involves, under the effect of phosphoglucoisomerase, as the name suggests, isomerase catalyzes isomerization reaction, converting one aldose derivative into a ketose derivative. Okay. Now, what is the necessity of this reaction? See, here this particular reactive group is hemiacetal. Here it is CH2OH. At first position it is CH2OH. So it is just hydroxyl group. Whereas in glucose it is a hemiacetal. Now, in the next step, we will see that this particular pathway involves conversion of 6-phosphate to fructose 1,6-biphosphate. So this is a phosphofructokinase reaction. So in this particular reaction, the first carbon OH attached to the first carbon is involved in this reaction, and this particular OH group reacts with ATP. It attacks the gamma phosphoryl group, a nucleophilic attack on the gamma phosphoryl group of adenosine triphosphate, and this results into transfer of this terminal phosphate onto this oxygen, giving you phosphorylated sugar derivative. So fructose 1,6-biphosphate is synthesized in this particular reaction. If you remember, I have told you that this particular reaction, like hexokinase, is also catalyzed by enzyme which belongs to class transferase, which catalyzes phosphoryl transfer. All such enzymes are called, as a general name is given, kinases. All kinases always use ADP or ATP as their substrate, depending upon whether ADP is used as substrate to synthesize ATP, or ATP can be used as substrate and it can be broken down and phosphate group can be transferred as seen here in onto the substrate. So because they use ATP, they always require magnesium ion because it stabilizes ATP, forming Mg-ATP complex. The structure of adenosine triphosphate, you can see magnesium ion sitting here. It binds with the oxygen which is having negative charge, the alpha phosphoryl group of oxygen of alpha phosphoryl group, and the negatively charged oxygen of beta phosphoryl group, stabilizes this. The gamma phosphoryl group is exposed, so OH group will make nucleophilic attack here and it will be cleaved and the phosphate will be transferred on to fructose 6-phosphate to synthesize fructose biphosphate. I mentioned that this is regulated by allosteric regulation. Let us just quickly see what is allosteric regulation. Allosteric is, allo is some other. So this particular enzyme has binding site for ATP other than the active site. Active site is the site where the substrate binds and the actual catalytic reaction takes place. That is called as active site. It is a small site. We will see later on when we study about enzyme. It is a small site, a cleft, a ditch-like structure onto which the substrate will bind and product will be released. Actual catalysis takes place. So apart from active site, allosteric enzymes can have other sites where substrate can bind and act as regulatory molecule, or where other molecules can bind and regulate the activity of enzyme. This particular enzyme is regulated by concentration of ATP, ADP, AMP, citrate and fructose 6-phosphate. Okay. So among all the molecules which I have listed just now, fructose 6-phosphate, AMP and ADP, they act as positive regulators. It means that when their concentration is high, the activity of enzyme will be increased by change in the structure of enzyme by allosteric regulation. So these molecules will bind, change the structure of enzyme such that it increases the rate of catalysis. In contrast to this, molecules such as ATP. So ATP has two binding sites. It will bind at active site at low concentration, but when the concentration is high, it will bind at active site as well as the regulatory site. And when it binds to the regulatory site, it indicates that ATP concentration in cell is sufficiently high. It is not necessary to produce ATP, and instead cell should decrease the rate of glycolysis such that now ADP will be available for further synthesis of ATP. Okay, so ATP will bind at allosteric site, decrease the rate of reaction. Similarly, citrate also acts as negative regulator. It has a binding site on the enzyme and this will decrease the rate of reaction catalyzed by phosphofructokinase. So what is the reaction? Okay, This is the reaction, conversion of fructose 6-phosphate to fructose 1,6-biphosphate. Now, fructose 1,6-biphosphate is acted upon by the fourth enzyme, which is called as aldolase. Aldolase brings about cleavage. So, it it is a 6 carbon sugar. It is cleaved. You can see here, it is cleaved here to give two carbon derivatives. This part is converted into dihydroxyacetone phosphate because it has this first carbon, which was carbonyl group in the linear structure. So this is the ketonic group. So this becomes dihydroxyacetone phosphate, and the other three carbons, they are converted to glyceraldehyde 3-phosphate. Okay. So aldolase belongs to enzyme class which is known as lyase. Lyases bring about cleavage. We will see later on enzyme classification. So lyases bring about cleavage, and here this molecule is broken down into two halves to give dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So in this reaction, we have seen it is a catabolic reaction. So degradation takes place in this step. Okay. So wherein a 6 carbon sugar is converted into two 3 carbon derivatives. Okay. Now, the next reaction can use glyceraldehyde 3-phosphate as substrate. But dihydroxyacetone phosphate cannot be used, and it needs to be converted to glyceraldehyde 3-phosphate. So, in the next reaction, we will see, which is catalyzed by enzyme triose phosphate isomerase. Again, isomerase, which will convert, as this isomerase is a triose isomerase. We have seen hexose isomerase before. So this triose isomerase converts one 3 carbon sugar derivative to other 3 carbon sugar derivative. Okay. So what is the reaction? Again, in this reaction, it is opposite to whatever isomerase we have seen before. The phosphoglucoisomerase converts aldose sugar to ketose sugar. This is converting ketose sugar derivative to aldose sugar derivative. So converting dihydroxyacetone phosphate, which has a ketonic group here, to glyceraldehyde 3-phosphate, which has aldehyde group here. Okay, how does this reaction take place? Now, when this reaction is taking place, what is happening? This, okay, phosphate is transferred onto this. Okay, so this CHO gets converted to CH2OH. Okay, now although I am saying that, you must be thinking that this is present at position 3 and this is present at position 1. Now the numbering will change. Okay, so numbering will change and dihydroxyacetone phosphate will be converted to glyceraldehyde 3-phosphate. Now, this reaction helps. The pathway to use all the 6 carbons can participate in the reaction in this way, because dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate. Now you will have 2 molecules of glyceraldehyde 3-phosphate which are going to participate in the next reaction. Okay, so we come back to the first slide. So we have acetone phosphate conversion to glyceraldehyde 3-phosphate. With this, the preparatory phase is over. The next reaction is catalyzed by enzyme 6th reaction, glyceraldehyde 3-phosphate dehydrogenase. So reaction number 6 catalyzed by glyceraldehyde 3-phosphate dehydrogenase. Now, as the name suggests, it dehydrogenates, removes hydrogen. So this is where oxidation takes place in glycolytic pathway. So 2 NAD molecules are converted to 2 NADH plus H plus in this particular reaction, and you can see glyceraldehyde 3-phosphate here is converted to 1,3-biphosphoglyceric acid. Now, after this step, you have to remember every step takes place twice, because we are starting now with 2 molecules of glyceraldehyde 3-phosphate. So every reaction will be taking place twice. Okay. So let us see the details of this reaction in the energy yielding phase. First reaction. So this is the structure of glyceraldehyde 3-phosphate as phosphate group at the 3rd position. Now, inorganic phosphate participates in the reaction. Okay. a correction needs to be done here. I should write 2pi here because two molecules of glyceraldehyde 3-phosphate are participating. So, two molecules of inorganic phosphate will be reacting, and aldehyde group at the first position is phosphorylated. Can you see here? Okay. So this phosphate, it is not obtained from ATP as we have seen in previous two kinase reaction. Here inorganic phosphate will directly react with the aldehyde group, and you will get a high energy compound 1,3-biphosphoglyceric acid. Now why am I calling this as a high energy compound? Because this molecule upon hydrolysis has ability to transfer phosphate group onto ADP so as to synthesize ATP. Okay, so this is known as high energy phosphate bond, which is synthesized by this particular dehydrogenase enzyme. Now the next reaction, that is the seventh reaction, involves synthesis of ATP. This is the first reaction of energy yielding phase which produces ATP. Now, since two molecules are reacting everywhere, here you can see that two molecules of 1,3-biphosphoglyceric acid are going to donate the phosphate group to ADP so as to synthesize ATP. ADP has two phosphate groups if you remember the structure. Now, the gamma carboxyl group will be synthesized by transfer of the phosphate group from a phosphorylated sugar derivative onto ADP so as to synthesize ATP. This particular reaction is example of substrate level phosphorylation. A reaction where ATP is synthesized. There are two different mechanisms by which ATP is synthesized. One is substrate level phosphorylation as seen here. The second mechanism is oxidative phosphorylation, which takes place during respiration. Okay, so this is substrate level phosphorylation, synthesis of ATP wherein phosphate is transferred directly from a high energy phosphorylated derivative onto ADP so as to synthesize ATP. The reaction is catalyzed by kinase. Now, what is the speciality of this particular kinase, phosphoglycerol kinase? This is the only reaction in glycolytic pathway which is catalyzed by kinase and is a reversible reaction in the cell. There are four different kinases present in glycolytic pathway. We have seen two kinases already, hexokinase, phosphofructokinase. Due to different concentrations of metabolites, these, although all enzymes catalyze reversible reactions, under cellular conditions, that is, the concentration of metabolites will decide the direction of reaction. So under cellular conditions, the concentrations will be such that in cell, the first two kinase catalyzed reactions, hexokinase and phosphofructokinase, are irreversible. They are unidirectional. Okay, take place in the direction which we have seen just now in glycolytic pathway. This is the only kinase enzyme which catalyzes a reversible reaction, and we will see later on a pathway which is known as gluconeogenesis, which involves synthesis of glucose from different molecules, amino acids, pyruvic acid, phosphoenol pyruvate, etc. So, reversal of glycolytic pathway takes place. This reaction is reversible. The other three reactions are not reversible, and three different enzymes are used in gluconeogenesis. We will see later on the pathway of gluconeogenesis, wherein I will mention about this enzyme again. What you have to remember is this is the only kinase reaction which is reversible under cellular conditions. Only kinase reaction in glycolytic pathway which is bidirectional and not unidirectional. Okay, can you see here? This is a unidirectional arrow. Other reaction is, this is reversible reaction. This is a unidirectional arrow. Okay, irreversible reaction. This is bidirectional. So all other reactions you can see, glyceraldehyde 3-phosphate dehydrogenase, this is also should be a bidirectional arrow. This is also a reversible reaction. Then this is a unidirectional reaction. Okay. So now let us go back to this reaction. Okay. So this is how ATP will be synthesized. The next reaction is phosphoglycerol mutase reaction. Mutase is a type of isomerase enzyme. So it converts one isomer into another isomer. What is taking place here? The phosphoryl group which is present at the third position is shifted on to second position. So 3-phosphoglyceric acid is converted to 2-phosphoglyceric acid. Can you see this has phosphate group attached to the second position here? Okay. So this particular isomerase just shifts the position. So it is a structural isomer, 3-phosphoglyceric acid and 2-phosphoglyceric acids are structural isomers of each other. What is the difference? Molecular formula is same. What is the difference? Position where the phosphate group is present. Here it is present on the third carbon. Here it is present on the second carbon. Okay. So this results into synthesis of 2-phosphoglyceric acid. Now, this actually is important reaction. It aids synthesis of phosphoenol pyruvate because it frees this third carbon. It is getting converted to CH2OH. Next reaction is catalyzed by enzyme enolase. Enolase catalyzes reversible reaction. In glycolysis, it will catalyze dehydration, removal of water molecule. How is the water molecule removed? You can see here this is the third carbon which has OH group attached to it and the second carbon has a hydrogen attached to it. Can you all see this? Okay. So a water molecule is produced using OH from the third carbon and H from the second carbon. This will result into freeing one valency each at second carbon and third carbon. So what will happen now? Introduction of a double bond. So these two carbons now have one valency free each when water molecule is removed like this. So they form a double bond. Okay. So a double bond is introduced here to synthesize one more high energy compound, phosphoenol pyruvate. This is again a high energy compound. Hydrolysis of phosphoenol pyruvate results into synthesis of ATP. We'll see in the next reaction. Now, this reaction, enolase reaction, also requires magnesium ions. For this particular reaction, magnesium ions are required so as to stabilize the phosphate in this particular reaction. Okay. Now, this is the last reaction of glycolytic pathway, reaction which is irreversible, catalyzed by enzyme pyruvate kinase. A kinase again, this kinase synthesizes ATP from ADP. Magnesium ions are required for this reaction. What happens here? This high energy phosphoryl group is transferred by phosphoenol pyruvate onto ADP so as to synthesize ATP. Okay. Again, this is a substrate level phosphorylation. Takes place twice in the cell. So 2 ATP molecules will be synthesized and the product of glycolytic pathway, pyruvic acid. Now, pyruvic acid is a 3-carbon derivative. So we started with a 6-carbon sugar. And what is the end product? A 3-carbon derivative, CH3CO COOH. Okay, will be synthesized at the end of the pathway. So let us go back to the first slide. So we have seen that first 5 reactions are of preparatory phase. The next 5 reactions include energy yielding phase where 2 different steps synthesize ATP. So 2 ATP molecules synthesized here, 2 here. Net ATP synthesized is, how many ATP molecules are synthesized in this pathway? Can you give answer?
4 molecules.
4 molecules of ATP are synthesized. But in the preparatory phase, you can see 2 molecules of ATP are being utilized. So what will be the net gain of ATP at the end of the pathway?
2 molecules.
2 molecules. So 4 minus 2. 2 molecules of ATP will be synthesized. Okay, so this is the net reaction. 6 carbon compound glucose reacts with 2 ADP molecules plus 2 inorganic phosphates plus 2 NAD plus gives 2 pyruvic acid molecules, which are 3 carbon derivative, 2 ATP molecules and 2 molecules of NADH, and 2 H plus will be generated. Okay, so with this we have finished with glycolytic pathway. Okay, now these are few of the images which I have used during pathway, and these are references for glycolytic pathway. So Leninger Principles of Biochemistry by Nelson and Cox, and Outlines of Biochemistry 5th edition, okay, by Kohn and Stump. So these are the basic references which you can use for glycolytic pathway.